Hi, I'm Belinne, and I'm going to be talking about morphometric approaches as a tool to distinguishing between three cryptic species of Myotis bats. To begin, cryptic species are difficult to tell apart in general. However, this becomes an issue when the species in question have different protection statuses. For example, Myotis austroriparius is listed as least concern. However, Myotis septentrionalis is listed as near threatened. So when researchers are going out in the field trying to collect austroriparius samples, they're trying to avoid septentrionalis bycatch. Because these two species forage in different habitats, we hypothesized that they would have different wing morphologies, which would allow scientists to differentiate between them in the field. So our objective was to compare wing elements of the two morphologically similar bats, Myotis septentrionalis and Myotis austroriparius, to determine if these elements could be used to distinguish between the two species in the field. To begin, we collected specimens from the Natural Science and Research Laboratory and measured 13 different wing elements, shown in Figure 1. The forearm, thumb, which is this component at the top, the second metacarpal, the 4 components of the third metacarpal, 3 components of the fourth metacarpal, and the 3 components of the fifth metacarpal. We measured each individual twice, and we measured the individuals in a different order for each trial. Once all of the measurements were taken, we constructed a data matrix, and we calculated the average of each wing element length. We separated the data by species and sex, and then conducted a multivariate analysis of variance, univariate analysis of variance, and a discriminant function analysis. So as you can see in Table 1, there were significant differences between species and sex in the forearm length and the fifth metacarpal length. The fifth metacarpal length. There were also significant differences between species in the thumb length, the second metacarpal, the third metacarpal, the first phalanx of the third metacarpal, the third phalanx of the third metacarpal, the fourth metacarpal, the first phalanx of the fourth metacarpal, and the first phalanx of the fifth metacarpal. Now, all of these univariate differences came together for one large multivariate difference. This multivariate difference is evident in the discriminant function analysis in Figure 2. So all of these dark blue and red dots represent austroriparius, and all of these light blue and pink dots represent septentrionalis. As you can see, there's no overlap between, so the wing morphologies are distinct between species. Most of this variation is due to the forearm length, the fourth metacarpal, and the third metacarpal. Because of our results, we concluded that wing elements can be used to differentiate between the two species in the field. Preferably, we would have had a larger sample size. However, septentrionalis samples are limited right now due to their protected status, due to their near threatened listing. Our findings still suggest a non-invasive field method of distinguishing between the two species as an alternative to DNA testing, which would be more time-consuming, more invasive, and would require biological samples. We do plan on conducting a third round of measurements and also measuring a third Myotis bat species, Lucifugus, so that we will hopefully have distinguishing characteristics for all sympatric Myotis in the southern United States. We would like to thank the Honors College Undergraduate Research Scholars Program, supported by the CH and Helen Jones Foundation. And we'd also like to thank Keith Garner of the Natural Science Research Laboratory. If anyone has any questions, I'd be more than happy to answer them now. Thank you.